So thank you everyone for being here today to listen to me. So my name is Vincent Bazinet. I'm from McGill University, more precisely from the Montreal Neurological Institute. And today in my presentation, I'll be talking about assortative mixing in microarchitecturally annotated brain connectomes. So when we study connectivity in the brain, usually what we do is we construct connectomes, which are basically a network of nodes and edges, where the nodes represent specific brain regions and the edges represent the relationship between these different brain regions. The goal of Connectomics has for a long time been to understand the physical relationship and functional relationship between brain regions and understand how they interact with each other, like through communication model, for example. And when we study connectome, for that reason, we're abstracting away all of the local biological information of the individual nodes in our network to focus on the more global relationships. But in reality, what we know about the brain is that every single brain region is unique. As I'm showing here, I'm showing an example of 8 brain maps that capture the huge heterogeneity of the brain across the cortex. And in my personal work, I'm trying to represent the brain by coloring. I'm trying to get an enriched representation of the brain by coloring every single node in our network with biological information that allow us to ultimately ask new questions about brain network organization. One specific question that is perhaps highly important for us is understanding very much the relationship between every single of those brain annotations or local microstructural attributes and brain connectivity. A lot of questions can be asked and a lot of question needs to be asked if we really want to understand what is happening, but in this talk, I'll specifically focus on the concept of homophily. So homophily is the idea that brain regions that have similar annotations, biological properties, are more likely to be connected than brain regions that have dissimilar properties. So the way I'm studying homophily in my work is using what is called the assortativity coefficient, which is basically a measure of the correlation between the annotation values of connected nodes. And the idea behind this measure is if you have a high correlation coefficient, it means that brain regions that have similar annotations are more likely to be connected. So in our work, what we did is we looked at 4 different connectomes from 3 different species, and we looked at a total of 14 different brain annotations on top of these connectomes, and we evaluated this assortativity coefficient. So as you can see here, every single one of our annotations were positively assortative, meaning that they all had homophilic relationships in the brain. But this is not really surprising given what we know about the brain in that it's a spatially embedded structure. And as a result, annotations in the brain are going to be spatially autocorrelated. What that means is that brain regions that are proximal in space will have similar annotations, whereas brain regions that are distant in space will are more likely to have dissimilar annotations. From the brain spatial embedding, there's also a wiring cost associated with connections. So what that means is that, what that results in actually is that most connections in the brain are short range. So if you combine these 2 ideas very simply, we'll obtain pretty much assortative or homophilic relationship between pretty much any annotation in the brain. But really what we want to understand here is specifically about the relationship between the topological organization of our connectomes and those brain annotations. So for that, we need to control for these global spatial constraints. And the way we do that is using what we call spatially autocorrelation preserving null maps. So these maps pretty much, I think a lot of you are familiar with those, but it's basically brain maps that we generate from the empirical brain map we're looking at, but we disrupt them so we can randomize them, and we obtain ultimately brain maps that have the same spatial autocorrelation structure in it, but any type of other relationship, for example, with the connectivity underlying the brain, will be disrupted. And then the idea is to look at the assortativity coefficient of these null maps and compare it to the assortativity coefficient of our empirical brain map. And so if we go back to our results, we can compare the assortativity scores we had for each brain map with the spatially autocorrelated brain maps. And then what's interesting afterwards is to compare those and compute, for example, a standardized value of each assortativity score with respect to these distribution. And so this is what I'm showing here. And now what's interesting is that it's not every single brain map that is significantly assortative above and beyond what we'd expect from the brain spatial embedding. It's a small subset. And this small subset is not random maps. These are very important brain maps that have been shown to be associated with connectivity in the past. So for example, if we look at gene PC1, T1, T2 ratio, and cortical thickness, they're all significantly assortative with respect to the functional connectome. And this is actually pretty consistent with the fact that they're organized along the principal gradient of functional connectivity. If we look at the macaque connectome, now we have T1, T2 ratio and neuron density, who are also significantly assortative, again, above and beyond what we'd expect from the spatial decorrelation of the brain. And this is actually, again, consistent with previous report, and it actually supports a lot of theories that suggest that cytoarchitecture in general is actually influencing the organization of cortical connections. We can think, for example, for those of you who are familiar with these theories about the structural model or the cytoarchitectonic type principle. So yeah, so after this main result, what we decided to do is investigate whether this homophilic properties in the brain are the same for short-range and long-range connections, right? And the way we did that is by thresholding our connectomes and removing all of the short-range connections in our networks and focusing on the long-range connections. And so the results are displayed here. So what we have here on the x-axis for each one of our 4 connectomes is the certain percentile of short-range connections that we have removed from our networks, leaving only the long-range connections. And on the y-axis, we have the standardized assortativity scores, again, standardized relative to the brain spatial embedding. And then what we can see is that as we remove more and more short-range connections, every single one of our brain annotation become less assortative with respect to, again, spatial correlation. And what's perhaps interesting is if you look at the top left graph here, so like in the human structural connectome, if we remove a large number of connections, actually what we see is that most of our annotation becomes significantly disassortative. And so what that means, basically, is that long-range connections appear to be linking regions that have attributes that are more dissimilar than we'd expect from the brain spatial embedding. And this is interesting because it might suggest that these long-range connections act or exist to kind of enhance the diversity of a brain regions and input and output. And this is actually relevant and related to other theories that have suggested similar things in the past. And we didn't stop there. We investigated this concept of assortativity and homophily even more by looking at heterophilic mixing in the brain. So this is, again, relying on the assortativity coefficient. But this time, rather than looking at a single annotation, we chose to combine 2 annotations together and compare their assortativity. So here the question we're asking is we're looking at individual regions in the brain, and we're asking, OK, if a region has a high value for an annotation X, is it the case that it's connected to regions of the brain that have a high value for another annotation, for example, annotation Y? And so to study this principle, we relied on receptor and transporter data, so density values from PET data that we have accessible freely, by the way, on NeuroMaps, our toolbox that we've created. And what we've obtained is this matrix here of standardized assortativity results. And I won't go too much into the detail for the sake of time, but you can see that there's a lot of significant relationships between pairs of neurotransmitter receptors and transporters that are related in a very particular way in the brain. So finally, the last thing I want to talk about is the fact that we can also look at homophily at the local level. So the way we did that is by computing a measure that we called the local homophilic ratio for each individual node in our network. I won't go into detail about the technique, but what's the important takeaway here is that we can actually look at which brain are the most assortative, which brain region are the most assortative and which one are the least assortative in our networks. And what we did for the structural connectome is we looked at these local homophilic ratios across all 5 of our annotations, and we computed the average and compared the average map of local homophilic ratio scores with neurosynth data. And so neurosynth is basically brain maps of probabilistic activation associated with specific cognitive terms. And then we grouped these terms into specific categories, and what we found is that the main takeaway of our work here was that brain regions that were involved in executive functions were most likely to be connected with similar brain regions to themselves, whereas brain regions that were involved in memory and emotion functions tended to be connected with nodes that were dissimilar to themselves. So this is particularly interesting because it's showing that these homophilic patterns in the brain might be related to functions in the brain as well. So yeah, so this was just a very short overview of everything we've done in this paper. You can come talk to me later on if you have more questions or want to hear about the rest of the things we've done in there. I'd like to thank my collaborators who helped me work on this paper, and thank you very much. And actually the code and data is actually available, so if you want to replicate that in your own analysis, it's all there. And yeah, thank you very much.
So we have time for one question. There are one question in the mobile app asks, how can different persuasion affect your assortative results?
That's a great question. These results are pretty consistent. Actually, like if you take a look at the paper, I think the first 3 supplementary figures we're looking across different parcellations. We have the Schaefer parcellation, we have the Camoon parcellation from the Lausanne group, and we've actually looked at different data sets of connectivity as well, and they're pretty consistent. We've actually looked at other spatially autocorrelated null models as well. Yeah, so you can take a look at the supplementary figure if you're interested, but yeah, the overall message is that it's pretty consistent.
Since our speaker responds very fast, so we have room for one more question. I'll let you just ask.
Thank you. Great talk. I had a question. So you mentioned the structural model from Helen Barbas's group, and I'm curious how you think about that, because in that model, which is mostly from monkey track tracing data, it's not just that cytoarchitectonically similar areas are connected more similarly, it's that the patterning of their connections feed forward or feedback into different layers of cortex is also patterned by whether an area is eulaminate or laminate or has granular or agranular cortex. So I'm curious how you've thought about kind of that level of detail in any of your data, which is hard to get with MR, but like maybe kind of masked by what you're looking at. Yeah,
that's a great question. So yeah, exactly, like the structural model is slightly more complicated than what I've tried to say here, but like we do have a figure in our work that is actually looking at the same thing I was looking at heterophilic mixing. We look at layers as well. So like from the big brain, like Conrad Wagsdale like did a very great job as like delineating every single layer in the structure and in the big brain data, and so we have cortical thickness for individual layers in the brain, and we evaluated whether like specific layers or like the thickness of specific layers is associated, like is homophilic or not across the functional and structural connectome. Unfortunately, we didn't find like precise results that were corroborating this theory of the structural model, but we still took a look at what it looked like in the human brain. But I mean, there are many things that might be not perfect there, so like we cannot say that the model is wrong or not, but yeah, we did investigate these questions as well.
Thank you.
Okay, thank you, Vincent. Due to time limit, we need to move to next speaker now. So next speaker